Okay, so there's a hidden problem with single-cell RNA-seq that most people don't talk about. To run single-cell experiments, researchers have to dissociate the tissue. That's right, they got to mash it up. And when you do that, you lose where those cells are located. And that problem led directly to spatial transcriptomics. For years, gene expression measurements have moved through 3 levels of resolution. First, bulk RNA-seq. And to be clear, bulk RNA-seq is still extremely useful for many experiments. It does a great job at measuring gene expression across bulk tissue samples, hence the name. Then came single-cell RNA-seq. So instead of averaging millions of cells together, researchers can now measure gene expression in individual cells. That was a huge leap, huge, massive leap, but it introduced a new problem, and that is around the single cell experiment. Specifically, researchers usually have to dissociate that tissue, like we mentioned previously, and when you do that, you lose where those cells originally live inside the tissue. That's where spatial transcriptomics comes in. Spatial transcriptomics measures gene expression while preserving the architecture of the tissue itself. So instead of only identifying the cell types, scientists can now see how cells interact, how they organize, and how they reshape entire microenvironments in a disease like cancer or neurodegeneration. It's a great reminder that in biology, location often determines function. And once you add that spatial context, the computational complexity explodes, which means the bottleneck is increasingly shifting towards data processing, data analysis, and visualization. But that's a topic for another day. By the way, these shorts are something of an experiment I'm doing. As you guys probably noticed, I'm using this really to explore the topics that happen across biotech and comp bio. If this is useful and you like the content, feel free to follow. Let me know what you guys would like to see next. I'm Dan Ryder with Bridge Informatics, and this was Informatics Insight. Thank you.